Hi, everybody. We're ready to begin Chapter 4, which is entitled Cellular Metabolism. And my plan for this particular lesson is to cover sections 4.1 through 4.3. And then we'll pick up with section 4.4 on the next Zoom tutorial or class meeting. So I'd like to begin by talking a little bit about this term metabolism. I think everybody has some innate sense of what that word kind of means. We sort of use it in our everyday language when we say things like, he's very thin. He must have a high metabolism, right? We often use it with respect to, using energy, or we think of it as maybe breaking down chemicals into smaller compartments or constituents, like during digestion. But in the cellular definition of metabolism, or the A&P version of metabolism, this word does refer to breakdown of bigger molecules into smaller ones. So digestion is a subset of metabolism, but it also includes the process of building bigger molecules from smaller, simpler ones. So I think we need to sort of expand our thinking of that word and not assume it just simply means digestive in nature. It could also be sort of the opposite. And we're going to talk more about that in just another minute or so. So I'm going to share the PowerPoint that you can find, of course, in Blackboard for Chapter 4. And we're going to start to think about these reactions as being composed of two different types, as I again alluded to just a few moments ago. The first term we're going to use, the first part of cellular metabolism, is referred to as anabolism. And this is what I was talking about a few seconds ago. This is the process whereby smaller, simpler molecules are put together, combined together chemically via bonding to make bigger, more complex molecules. And in that process, not only do we make a bigger, more complex molecule from smaller, simpler ones, but we also do this by removing water from the reactants, from the building block monomers here, so to speak. And this process of anabolism, whereby we're making the bigger molecules from smaller, simpler ones by removing water, takes energy, takes ATP, we call it. We'll define that toward the end of today's lesson. But it's the energy currency of our cells, this adenosine triphosphate, or ATP for short. And so you see in front of you here this chemical reaction, which has a special name, obviously. It's called dehydration synthesis. It is an anabolic reaction. And in this example, we're taking two monosaccharides, which you should have learned about back in Chapter 2 on the chemistry chapter, where you reviewed the major organic molecules, right? Carbohydrates, lipids, proteins, and nucleic acids. So here we've got two building block carbohydrates. They're called monosaccharides. And we're combining them anabolically in dehydration synthesis. The end result is the formation of a bigger, more complex molecule, as promised by the definition. And we are also making water as a result. So water is a byproduct of this process. Now, again, where did the water come from? Well, you got to go back into the reactants, in this case, the two monosaccharides. And you'll see here in blue where they've shown you where the one hydroxyl group here, the OH group coming off what we call carbon number 1 of this sugar. We usually number these carbons, if you remember again from Chapter 2, the apexes of these hexose sugars denote carbons. So there's a carbon 1, carbon 2, carbon 3, carbon 4, carbon 5, and here's the 6th carbon of this monosaccharide. So coming off carbon number 1 is an OH group, and coming off carbon number what? 1, 2, 3, 4, yeah, is another OH group, and we're going to note again the hydrogen from this particular OH group, which when combined with the hydroxyl group of the adjacent monosaccharide combines to give you the H2O. And look what happens. We form a link, a bond, don't we, between this sugar and this sugar. There's the bond right here. That oxygen right there is this oxygen right here. And then, of course, you see the 2 hydrogens, and the rest of the molecule is the same. But this is a disaccharide, isn't it? It's 2 monosaccharides that have been linked together thanks to dehydration synthesis. The other half of metabolism is called catabolism, and this is the one that you're probably more familiar with. Again, think back to the phrase, he's really thin, he must have a high metabolism. Okay, so this denotes the idea of taking bigger, more complex molecules and breaking them down into smaller, simpler ones. So it's sort of the reverse of anabolism, isn't it? Note, however, though, that this process it doesn't require energy, but actually releases energy. Okay, so it's a little different in that sense. It's producing energy. Now, a lot of the energy that's produced during this breakdown process, which is really a digestive process in and of itself, a lot of the energy that's given off by you right now, as you're sitting there watching this video, is in the form of heat. If you had a thermal camera and you took a picture of your arm or your body, you'd see heat coming off of it. And in the same way that if you're at a party and you're in a small room and there's a lot of people, right? It's hot in there. That's why, because people are giving off energy in the form of heat. In fact, this is, as it says here, about 60% of catabolic energy release is in the form of heat. The other percent, the 40-odd% remaining, can be used in the previous example we talked about in the previous slide of making bigger molecules from smaller ones, right? Anabolic. Our cells do that all the time. And as we'll talk more about in another part of this chapter, cellular work is taking place all the time as well. You've had a sneak preview of active transport. We talked about that in chapter 3, I think it was. Remember, this was where cells take compounds, molecules, ions, and pump them against a concentration gradient. Remember our example of the salmon swimming upstream, right? That analogy or metaphor? Well, that takes energy, doesn't it? And so that energy that cells use to pump ions across the membrane against their concentration gradient comes from the degradation of bigger molecules that we're digesting, really. Muscle contraction, you know, whether it's skeletal muscle or your heart, cardiac muscle beating, that all takes energy. You need to have energy for all sorts of cellular activities. There's sperm cell, right, swimming toward the egg, if you will, expending energy to power its flagellum. Another example of energy usage at the cellular level. That comes from catabolism. So in this example, we see, again, just the reverse of dehydration synthesis. This is called hydrolysis. If you look at that word and dissect it, hydro refers to water, lysis meaning to cut or to break apart. So here we're taking a disaccharide, we're incorporating water into the reaction, we're making smaller, simpler monosaccharides, of course, in this process. And where did that water go? The 2 hydrogens and the oxygen? Well, again, they're accounted for here, aren't they, in the various parts of the 2 monosaccharides. So we can sort of think of these 2 very important chemical reactions, one of which is anabolic, dehydration synthesis, and the other is catabolic, hydrolysis. It can take place in carbohydrates like we see here, or it can take place in other organic compounds too. Here we're looking, of course, at lipids or fats. And in this particular example, which is catabolic. We're taking glycerol, this compound shown here in blue. We're combining it with 3 fatty acids. Again, this should be a review from Chapter 2 Chemistry, where you learned about lipids and what they were made up of, and what the difference is between a saturated and an unsaturated fat. Remember all this? We're taking those 3 fatty acids and the glycerol, and in this particular example, we're moving the reaction from left to right, dehydration synthesis. We're making a bigger, more complex molecule. It's called a fat or triglyceride. And note, we're making 3 water molecules. Well, why 3 and not 1, like we saw earlier in the carbohydrate example? Well, again, look at what's going on. We're linking 3 fatty acids to the glycerol. 1 fatty acid is getting linked here, 1 here, 1 here. There's the 3 waters, right? And we're making a bond between each of the fatty acids and this glycerol molecule to create what's called a triglyceride or fat. That takes energy to do that. It requires ATP for a cell to make a bigger, more complex fat from smaller, simpler building block molecules. We could also talk about this as it pertains to hydrolytic or hydrolysis reactions, which remember are catabolic. In this example, we're taking the bigger, more complex fat, we're incorporating water into the reaction, and we're making the glycerol and the 3 fatty acids. We'd also end up making some energy as well. It's not listed here, but remember we talked about that. And we could go and talk about another organic compound, in this case, how are proteins produced? Proteins, remember, have building block molecules called amino acids. So if you want to build a protein, you have to start linking amino acids together. You have to do this via dehydration synthesis because it's anabolic. So in this example, we're taking 2 amino acids, we're linking them together. Of course, every time we do that, we make a water molecule. For each amino acid that gets added, we make a water molecule. We also create this peptide bond between the amino acids. Now, this is just the very beginning of making a protein. We'd have to add hundreds, if not thousands, more amino acids to make a fully functional polypeptide or protein. But basically, every time you link an amino acid, you're undergoing dehydration synthesis, and you'd be creating a water molecule too. And of course, we could reverse the reaction in the case of catabolic metabolism, where we're breaking the bond right here, incorporating water into the reaction, making our 2 simpler amino acids. So these reactions, dehydration synthesis and hydrolysis, are extremely important for catabolic and anabolic processes. And these reactions are going to employ special helper molecules, which are going to affect the rate at which you build up or break down organic compounds in a cell. And we're going to be talking next about these all-important things called enzymes. You've all heard of them. You associate them with digestion, don't you? You've heard of enzymes in your small intestine, perhaps. You'll be learning about that next semester. Pepsin is an enzyme produced in your stomach to break down proteins. So we typically think of enzymes in terms of their role in catabolic reactions, breaking down bigger molecules into smaller ones. And that's perfectly fine, and we should think of enzymes in that way. But I'm here to tell you that they're not only used to break molecules down catabolically, they're also important in anabolism, which of course, again, is taking smaller molecules and building up bigger, more complex molecules from them. So you got to, again, think about enzymes as being dual purpose, build up and breakdown. Important in helping those processes take place. So what are enzymes? Well, they are proteins. They belong to the protein family. They are reusable. That's kind of interesting. They're green in terms of being able to recycle them. Okay, they're reusable. Our cells can reuse them all the time. They are specific with respect to what substance they combine with and alter. Again, either building it up or breaking it down. And they help reactions go much more efficiently for a cell, much quicker as well. So they're just extremely important proteins. They help reactions. Without enzymes, many reactions would be unable to take place. It would be just too costly to the cell without enzymes. So here we're looking at how an enzyme kind of works. And I'll let you watch this like 1.5 minute YouTube video that will pretty much summarize what I'm going to talk about here next. We're going to depict this enzyme, this protein, as this kind of purple colored model, if you will. And I want you to make special note of this part of the enzyme called the active site. It's literally a three-dimensional region of this protein. Now, all molecules have three-dimensional conformations or shapes. You probably have never thought of this very much, but you have to start to think about these molecules having three-dimensional structures. Here's a good example. You all know what DNA shape, what the shape of DNA is, I think, right? If you don't, you should. It's a double helix, right? So it has a particular shape, and that particular shape affects its function. It's a structure-function relationship at the chemical level. And so proteins and carbohydrates and lipids and nucleic acids, those organic compounds, all have their own unique three-dimensional shapes or conformations, which affects their function. And there's no greater example of how this is so critical to the action and function of a molecule than looking at enzymes. So this three-dimensional region of the enzyme called the active site has a particular shape associated with it. That's all important in what molecules the enzyme is going to change or alter. And of course, you see in our example, we have two substrate molecules that are going to be impacted by the enzyme. Note, we're showing one yellow and one blue. We're giving them their own unique conformation or shape too, which they will have. And so what we're going to do is we're going to actually go from left to right here and talk about an anabolic process, whereby we're taking smaller, simpler molecules, and we're going to end up making a bigger, more complex product here at the end, aren't we? So how do we do that? Well, we use this special enzyme to allow the substrate molecules to fit into the active site of the enzyme. So the shape of the active site determines what substrates will fit into it. That's all important. Not any old substrate molecules can fit into this, right? Their shape has to conform to the active site shape. And so here we have what's called an enzyme substrate complex, whereby the substrates literally conform to the active site of the enzyme. There's a little bit of bonding changes and alterations that take place here, and we end up producing a new product, and that enzyme goes back to its original shape, unaltered, to be reusable. Remember we said earlier that these enzymes can be reused again and again and again. And so what's this guy ready to do? Well, it's ready to combine with more substrate, if more substrate is there, to produce more product, right? Now notice that these reactions are reversible. What this basically means is that, yes, we can talk about anabolic reactions here, dehydration synthesis reactions, left to right, but if we go from right to left, that is catabolic, right? That's hydrolysis. That's taking a bigger molecule, breaking it down into smaller, simpler ones. Enzymes are all important in that process as well. So I'll let you watch that short little video, which will again just sort of review what I just talked about. As I said a little bit earlier, we need to think of enzymes not only in digestive catabolic reactions, but they're also equally as important in anabolic reactions as well. Here we're looking at the conversion of a substrate, we'll call it number 1, into a different molecule, we'll call it substrate number 2. We're altering it thanks to the action of enzyme A. So substrate 1 fits into the active site of substrate of enzyme A, excuse me, and the end result will be the production of a different molecule. It's been altered in some way. We're going to then have that substrate 2 be altered to form a different substrate, number 3, thanks to the action of a different enzyme. We call it enzyme B. Substrate 3 being converted into a different compound by a different enzyme, enzyme C. And let's take it one step further and talk about a fourth enzyme, we'll call it D, necessary to convert substrate number 4 into some desired end product by the cell. So to review, we've started with a particular substrate number 1. We've converted it into a number of different other compounds or substrates. But at the very end, we have produced, the cell has produced some desired end product that it needs or wants. This occurred again thanks to the action of 4 different enzymes, right? Each converting a particular substrate. And until finally we get to the end result here of the product we want, the cell wants. In reality, it's not 4 reactions in your cells, it's hundreds and hundreds and hundreds of different reactions, all catalyzed by specific different enzymes. It gets very complicated. But if you can understand this basic, simple example, I think you're fine. Now I want to introduce this concept of how a cell produces just enough product to use. How does it know not to produce too much product? Because there's no point in making more than you need, right? That would be very inefficient for a cell to do that. So how many metabolic reactions are sort of regulated in our cells is through a process referred to as negative feedback. Now you've heard of this. We talked about it in chapter 1 as it pertained to homeostasis, right? The maintenance of, say, body temperature. This is negative feedback as it applies to chemical pathways. Okay, so this is a little bit different, but it operates on the same premise of negative feedback. In the same way that your body temperature, once it reaches a certain point, you begin to sweat, let's say when you get too hot. Once you start to cool down, you stop sweating, right? You negate the action of that temperature rise. Well, in this particular instance, when the level of the product gets to a certain point and the cell can't use any more of it or doesn't need any more of it, the level of the product has an inhibitory effect on the first enzyme of the reaction, which is enzyme A, right? Our beginning enzyme that converted 1 into 2. If this rate-limiting enzyme converts less substrate 1 into substrate 2, what's going to happen to the level of substrate 3, 4, and ultimately the product? So it's going to go down because it's limiting the conversion. It's not allowing as much of 1 to get converted into substance 2 or substrate 2. These other enzymes, B, C, and D, are not impacted here at all. They'll convert as much as they can. But if we can influence the first reaction in the process, which is what we're doing here, and convert less of 1 into 2, the end result is going to be less product being made. Now, when that product level gets low, what do we want to do? And the cell says, I need more product. Well, there's going to be sort of an inhibition of the inhibition, right? And this rate-limiting enzyme is going to be given the green light. Okay, convert as much substrate 1 into 2 as you can. What's that going to do? Ultimately, to the product level, it's going to bring it back up. So it's it's classic negative feedback, but at the enzymatic cellular reactionary level, if you will. Here's a molecule we talked a little bit earlier about, ATP, or adenosine triphosphate. I talked about it as the energy currency of the cell, of your cells, my cells, every animal cell, even bacterial cells. And so, again, I think chapter 2 may have talked a little bit about it or introduced it, but just to kind of go over the chemistry for just a second, you can see where the molecule gets its name, the triphosphate, pretty self-explanatory, 3 phosphate groups. Note these squiggly wavy lines we'll talk more about in a few minutes. These are chemical bonds, but they're unstable chemical bonds. This is adenine. We'll talk about it coming up when we discuss more about DNA, but it may sound familiar to you. It's one of the nitrogenous bases of nucleic acids. This is called a ribose sugar, which you may recognize from RNA. And so it's bonded to this adenine. This is a covalent bond here. And then we have another bond between the ribose sugar and the 3 phosphates. So collectively, this whole thing is adenosine triphosphate. This is the molecule, this is the energy currency molecule our cells are making as we speak as a result of hydrolysis reactions, catabolic reactions going on right now in you and me. We know from an earlier segment of chapter 3 that one super important organelle of our cells is the mitochondrion. That's the single organelle. If I'm talking about plural, I'd be using the term mitochondria. So it's within the mitochondria of our cells, sometimes affectionately referred to as the powerhouse, right? That this ATP is being made all the time. Critically important process. We have to create this ATP, this energy, because without it, our cells will die. And so here we're talking about this process of creating ATP. When we take ADP, adenosine diphosphate, and we add a phosphate group, that's what we're showing here. We introduce a phosphate group into the reaction, and we're adding obviously a 3rd phosphate, creating our ATP. Because almost as quickly as we make it, our cells are going to break it down. We're going to pull a phosphate group off. That phosphate group is going to help in cellular work, which we'll talk about that process a little bit later. But we're going to make ADP, and then we have to resynthesize the ATP by adding more phosphate. So we're either pulling phosphate groups off, or we're adding more phosphate groups on as we recycle this ATP molecule. So let's talk about how we take adenosine diphosphate, add a phosphate group to it, and make ATP. This is the process called cellular respiration. And it'll be the primary topic we're going to talk about in our next lecture. So I'm going to stop here. This next section will begin section 4.4. So we've just finished 4.1, 4.2, and 4.3. Okay, see you in a few minutes in the next section.